Well, good afternoon, everyone. So last time we already discussed the citric acid cycle, and we know how important it is and why it is important. And we basically give you a big picture for the whole process, right? And we know that the purpose for this citric acid cycle is not really generate much ATP. Instead, it generates more electron carriers, and those electron carriers will further enter the oxidative phosphorylation to generate more energy. So that we will discuss in chapter 18. So we talked about two steps, the first two steps of the citric acid cycle. One of them is the condensation reaction to combine the oxaloacetate and acetyl-CoA to form the citric-CoA, and eventually to form the citric and release CoA. So this reaction is an ordered sequential displacement that you need to know. The second step is isomerization. So it isomerizes citrate to isocitrate. The reason for that is because citrate is not stable by having the hydroxyl group and the carboxylic group attached to the same carbon. So it's not stable, right? So after isomerization, it becomes stable. So the two enzymes catalyze these two reactions, so you need to know them. So the first one, the citrate synthesis, basically is an induced form, is a induced fit. And the second enzyme, the aconitase, is an iron sulfur protein, but it's a non-heme protein. Okay? So that is last time we discussed. So today we're going to continue talking about the citric acid cycle, finish the rest of the steps. Okay. So step 3 is an oxidation reduction reaction, basically. So it's going to oxidize the isocitrate to be alpha-ketoglutarate. And the oxidizing agent is NAD+. So NAD+ itself will be reduced to be NADH by gaining a hydride, right, with an electron. So this actually also a two-step reaction shown here. The oxalosuccinate is an intermediate, and eventually it will remove a molecule of carbon dioxide to form this alpha-ketoglutarate. So this is an important reaction because this is the first reaction that release a carbon dioxide. It means this 6-carbon molecule becomes a 5-carbon molecule. You're losing a carbon. And this is the carbon that is removed or oxidized. Okay? So be careful here. Since we have this isocitrate, you have to know which carbon is removed. Okay? That's an important thing, because later on when we talk about isotope label, so we need to know how to track the carbon in this citric cycle. That is step 3. The 6 carbon becomes 5 carbon and you release the carbon dioxide. The step 4 is another reaction to release another molecule of carbon dioxide. So the alpha ketoglutarate is converted to succinyl-CoA through an oxidative decarboxylation. It is also a redox reaction. In this redox reaction, the first carboxylic group is oxidized to carbon dioxide and then removed from the alpha ketoglutarate. And this reaction is catalyzed by alpha ketoglutarate dehydrogenase complex. If you still remember, before enter the citric acid cycle, the pyruvate is converted to be acetyl-CoA. So that reaction is catalyzed by a pyruvate dehydrogenase complex, right? So that complex contains 3 enzymes, 5 coenzymes. So this alpha ketoglutarate dehydrogenase complex is very similar as the pyruvate dehydrogenase complex. It also contains 3 enzymes and even have a same enzyme as the pyruvate dehydrogenase, the E3. So that is what you need to know. And of course, which one is reduced, which one is oxidized. NAD plus again is reduced. Oh, here I should write NAD. Actually here. So NAD plus is reduced to be NADH. That's step 4. So step 5 generates ATP from ADP. This is the only reaction in this citric acid cycle that generates ATP. Remember, only one molecule of ATP is generated. That is this step. So the succinyl-CoA reacts with the ADP and the phosphate ion to produce ATP and also form the succinate and then release CoA. So this reaction is catalyzed by succinate synthesis. Succinate synthesis is a dimer. It contains 2 alpha beta subunits. So the general structure, you don't need to know exactly details. Okay. And the succinyl-CoA is an energy-rich cell ester compound, right? So it's a cell ester compound with CoA. And then the cleavage of this cell ester bond of succinyl-CoA is coupled to the phosphorylation of ADP or a GDP. If in the muscle, including the heart muscle and the skeletal muscle, so it will use ADP. If it's in the liver, it will use GDP, the guanine diphosphate, right? Adenine diphosphate. So this enzymes catalyzed, and by the way, so if we use GDP, it will form GTP, is another form of energy. So the enzyme catalyze the reaction in the, using ADP or using GDP, they are isozymes. We learned before, right? They are the same function, but located in different organs. So about this enzyme, so we already say it's a dimer, contains 2 alpha beta subunits, and we do have mechanisms. we do have the mechanisms about the enzyme, the succinyl-CoA synthesis. Right, so histidine here plays an important role. First, the phosphate ion will attack this carbon, the carbonyl carbon. Oh, no, sorry, actually the ketone carbon, right? So it will attack this carbon to attach. Then the CoA will be released. And then the histidine here plays an important role like a transporter, basically. So it will grab this phosphate ion to itself. This phosphate ion will be removed from this succinyl compound or succinyl phosphate. So now the phosphate ion connects to the histidine. So further, the histidine will like a swing arm. So from here becomes here. It changes the position. And it will transfer, or it will, pass this phosphate ion to ADP and then form ATP. So it grabs phosphate from this succinyl phosphate compound, and then it passes this phosphate ion to an ADP to form ATP. Okay, so then another cycle starts. So for this mechanism, you need to know the role of histidine. It's like a transporter, right? Swing transporter. Grab phosphate and then transfer it to ADP. And also you need to know the structure for the enzyme. So that is for step 5. So the rest of steps 6, 7, 8, we're going to combine them together to discuss. So overall, these 3 steps are the regeneration of the oxaloacetate. So remember the first step, we have the acetyl-CoA react with oxaloacetate to form the citrate, right? So, and it's a circle. So the last step, you actually regenerate oxaloacetate to enter another citric acid cycle. So let's see how can this regeneration be completed. So starting from the succinate from step 5, we have this structure here. So these 2 hydrogens in the carbon 2 and carbon 3, they will be removed. Or we say they will be transferred to FAD to form FADH2. So apparently, this reaction should be an oxidation reaction, oxidation reduction reaction. So the FAD will accept the 2 hydride to form FADH2. So FAD is reduced, and this succinate will be oxidized, so to form this carbon-carbon double bond. So that is a fumarate, okay? So succinate is oxidized to be fumarate. FAD is reduced to be FADH2. So here you may got a question. So before, when we have oxidation reduction reactions, we always use NAD+, or mostly we use NAD+ to accept hydride to form NADH. Right? We use NAD+ as an oxidizing agent. So why here we use FAD instead of NAD+? So the reason is actually because the free energy change is not enough to reduce NAD+. So in order to reduce NAD+, you need more energy change. However, this reaction doesn't have that much. So you can only reduce FAD, okay, which require less energy. So about the enzyme that catalyzes this reaction is the succinate dehydrogenase. So again, when we say dehydrogenase, usually it catalyzes, oxidation reduction reaction that you need to know. So this enzyme is also an iron sulfur cluster protein. So far we have learned two of them, right? One is a cognitase. This succinate dehydrogenase is another one. So very interesting. This FADH2 being formed from FAD. So it will stuck with the enzyme, and it will not leave. It will stuck with the enzyme and directly transfer the 2 electrons. It just accepted. It will transfer these 2 electrons to the iron sulfur cluster of that enzyme. And this iron sulfur cluster will pass these 2 electrons to a coenzyme Q. This Q will further enter the oxidative phosphorylation, which we will discuss more details in chapter 18. Okay, so actually chapter 18 we're going to review this enzyme again. Very interesting. And that's why when you have the reaction occur, so you put a EFAD, means the enzyme will bind to the FAD. And when you produce your product, it's an EFADH2, means the FADH2 still stuck with the enzyme. So step 7 is a hydration reaction, which you add a molecule of water to form the hydroxyl group and a hydrogen. So step 8 is another oxidation reduction reaction. So this time we do use NAD+. So the NAD+ is reduced to be NADH, and the molet is oxidized to oxaloacetate. So this oxaloacetate will enter another cycle of the citric acid cycle. So what you need to be careful here is the delta G. The delta G standard for this oxidation reduction reaction actually is very positive. It's positive 29.7. So which indicates theoretically it should be a non-spontaneous reaction, right? Always say thermodynamically unfavorable because positive delta G. So but why this reaction can still occur? If you look at the whole reaction, the whole cycle, if you look at the whole cycle, when you form oxaloacetate, it will enter another circle. It will get consumed. You need to use this oxaloacetate, right? It will be used, which means the concentration of your product will be decreased. And also for the NADH, it will enter the oxidative phosphorylation. So both these two products will be used for later reaction, which means the concentration of these products will decrease. According to the Le Chatelier's principle, when the concentration of products decrease, so your equilibrium will shift right? Shift to the right. So that drives this reaction occur forward. And since it's in the biological system, so this is going to be continue and continue. So that is what drives this reaction to take place forward, make a thermodynamically unfavorable reaction occur. Okay, just be careful how to explain this. So that's all the 8 steps for the citric acid cycle. So a couple of enzymes involved, especially those 2 with iron cluster enzymes, and also some oxidation reduction reactions involved. So you need to know. So now let's talk a little bit about the isotope label. So when we started chapter 16, the glycolysis, we learned how to use isotope labeled carbon to trace where are the carbons goes, right? Where are the carbons goes eventually in the carbon dioxide or in the pyruvate. That's for glycolysis. So for citric acid cycle, we can use similar way to track the carbon. For example, we know that the acetyl-CoA is converted from pyruvate. If I label the methane group in the pyruvate, this carbon H3, right? I label this carbon. So eventually, where is this carbon goes? It will be removed as a carbon dioxide, or it will stay as an oxaloacetate. So let's track to see where it goes. So you label this carbon, and you have this old oxaloacetate, right? So this is acetyl-CoA. So here I forgot the star should be here. So the carbon is labeled and oxaloacetate. So a condensation reaction is formed. So the acetyl group is added to the oxaloacetate, right? So this is your, excuse me, this is your methane group. So the carbon 14, isotope means carbon 14. This carbon 14 is here. So further isocytrate, no change, but still this carbon here. So further, you remove a carbon dioxide. Okay, remove a carbon dioxide here. So the carbon dioxide that is removed should be the middle one. So actually, it's the one from your oxaloacetate, right? From your oxaloacetate. So the labeled carbon is still here, right? Not changed yet. So now, further, this alpha-ketoglutarate will remove another carbon dioxide. I don't know why I didn't write carbon dioxide, but it will remove another carbon dioxide. Oh, actually here. So that should be this carboxylic group, right? So it will be removed. It forms the the succinyl-CoA. So 2 carbon dioxide are removed, but none of them is from your labeled carbon. So is the labeled carbon still here? Labeled carbon still here. Is the succinyl-CoA. Is a labeled succinyl-CoA. Okay. So now further, succinyl-CoA becomes succinate. So pay attention to the structure of succinate. If you notice, we have carboxylic, CH2, CH2, carboxylic. So it has a symmetric structure, right? It has a symmetric structure. So which means, although I labeled this carbon should be here, this carbon, but you don't know in the next. So because I can name this in number 1, number 2, number 3, number 4, or I can name them number 1, number 2, number 3, number 4. Basically, I say these two carbons are the same position, right? So although I labeled this carbon here, but the carbon can occupy any position from these two. Can be this carbon, can be this carbon. Or we say in the pyramid, this carbon can come from this carbon, or it may come from this carbon. So here you can still differentiate it here. But in this fumarate, you don't know where this carbon comes from here or from here. These two are the same equation, the same position. Okay, so that means in fumarate 2, actually starting from the succinate, I don't know which carbon is labeled. Maybe this one, maybe this one, because they are the same position. Same thing for fumarate. So which carbon is labeled? This one or this one? I don't know. Maybe half-half, right? So same thing. When you have morlate, still these two can be both labeled. This carbon or this carbon. So even when you form oxaloacetate, you still don't know is this carbon or this carbon. So both these two carbons can be isotope labeled, right? Both these two can be isotope labeled. But when you enter this oxaloacetate, enter another citric acid cycle, what will happen? These two will be released, right? These two will be both released as carbon dioxide. So which means in the second round of citric acid cycle, there will be no isotope labeled carbon because that will be removed from the cycle. Okay, so but in the first round, the isotope labeled carbon will stay in the oxaloacetate in these two carbons. So that is for the isotope labeled carbon, how to trace where it goes. Okay, there's a similar problem in your workbook, so you can work on that problem and get a better idea. All right. So that's basically for all for chapter 17. The rest part for 17.4 is really kind of give you a big picture for the metabolism, right? So how these relationships, how we say this citric acid cycle is a hub. So pyruvate connects with glycolysis and then further they form glutamate, pyrene, heme, all this. So overall reaction, right? So again, glycolysis and the citric acid cycle, the oxidative phosphorylation here, right? So, and the fatty acid oxidation, fatty acid. So we're going to discuss for fatty acid at the end of the semester. So eventually the products will be acetyl-CoA too. So actually the acetyl-CoA entered in the citric acid cycle comes from two parts. One is from glycolysis, the other is from the metabolism of fatty acid or fatty acid oxidation. And then the NADH and the FADH2 will enter the oxidative phosphorylation. Okay, so it's really like a big big hub. Here, this graph, you can. So probably when we finish chapter 18 and chapter 22, we will go back, check this chart. It makes more sense. Okay. So really for 17.4, you don't need to worry too much. So you won't be tested. There will be no question related with that part. So only 17.1, 17.2, and the introduction part will be in your exam. And I'm not sure whether you checked the iCollege. So I changed the contents for exam 3. So exam 3 will only covers chapter 11, chapter 15, and chapter 16. Chapter 17 will be in exam 4. Okay. These problems are pretty straightforward. You can go back and find the answers. Oh, by the way, so the location for the succinate dehydrogenase, I think I put on the note, but I didn't. Okay. Yeah, so I think I forgot to put on the note, but here. So the succinate dehydrogenase actually is located in the inner mitochondrial membrane. So that is a very special location. All the others are located in the outer mitochondrial membrane. Okay, and a special enzyme. The others, I don't think there's problem, right? So here, this question about approximately how many ATP GTP equivalents will be produced for 1 turn of citric acid cycle. Actually, there should be 10 of them because that's probably when we finish chapter 18, it makes more sense. Later on, the NADH and FADH2 will produce about 9 ATP. If you still remember, in the beginning of this chapter, I actually mentioned that. Right, so this NADH and FADH2 will generate 9 ATP when they enter the oxidative phosphorylation. And the citric acid cycle itself will generate 1 ATP. So overall, it will generate 10 ATP for 1 round. Okay, that is for this problem. So here the citric acid cycle is inhibited under high energy conditions. It means if you have high energy, you don't need create more, right? So it will be inhibited. Same thing for glycolysis, actually. If you get high energy, you get inhibited. That's why, remember, within the regulation of glycolysis, ATP usually is an inhibitor. That's pretty straightforward. The net equation for citric acid cycle, you don't need to remember exactly this equation. What you need to know is removed 1 FADH2, 3 NADH, and 1 here wrote GTP. If you write ATP, that's also right. Okay, that's our important things you need to know. So that's all for chapter 17.